Good afternoon, everyone. We will get started now. So today is lecture 6, which is about cellular reproduction. It is the first lecture in the second half of the course. So let's recap some of the things that you should know by now. We learned from the cell theory that all living things are made up of cells and that they have hereditary material that they can pass on to the next generation. And then one of the other points in the cell theory is that all cells comes from pre-existing cell. So you are going to have one cell here, and we call this the parent cell. Okay, so in humans, the parent cell is going to have 46 chromosomes, 23 from mom and 23 from dad. And what happens is, before the cell divides, it is going to double all its genetic material. Okay, so 46 becomes 92. Okay, and the process by which this happens is what we call DNA replication. Okay. Once the DNA has been replicated, then the cell is ready to split, and you end up having two genetically identical cells. Okay, so these are two genetically identical daughter cells. Okay, and this process is called mitosis. DNA replication and mitosis is the focus of today's lesson. Okay, and again, I assume you have watched the recordings, and I'm gonna just give you an overview, a recap of some of the key points. So first, we have a table here that has all the major key scientists who have contributed to the discovery of DNA, as well as properties of DNA, and mechanisms related to how DNA is replicated. So let's start with the first one. First one is Chargaff, and he basically, let me zoom in a little bit, basically discovered that the DNA in the DNA, okay, in any DNA, the percentage of A, or we can say the number of adenine, okay, or A, always equals to the number of thymine. Okay, and the number of guanine, which is G, always equals to the number of cytosine. Okay, this is not surprising because we know that A pairs with T and G pairs with C. So you would expect to have the same number of A as you would for thymine, and same number of G as you would for number of C. Okay, so the Chargaff's base pairing rule, okay, DNA base pairing rule, DNA base pairing rule is that A pairs with T while C pairs with G. We know this already. Okay, so sometimes this is also called the Chargaff's rule. All right. Rosalind Franklin. She used X-ray to study the structure of DNA. And what her data shows is, her data suggest, we should say suggest, DNA is a double helix. Okay, so we're all familiar with the twisted ladder, right? So she was not able to finish her work because she died from ovarian cancer, probably due to the exposure of X-ray. Back in the days, they didn't have as much protection as they should while working with X-ray. So the next comes Watson and Crick. So they built the first DNA double helix model based on Franklin's data. Okay, so they actually have a scaled model. If you Google Watson and Crick, there is a very famous picture that is in almost every textbook that show them, examining the 3D DNA double helix model that they built. They then went on to win the Nobel Prize for that. But again, the data was based on what Rosalind Franklin has discovered. Okay, we have Hershey and Chase. Hershey and Chase, they use bacteriophage. Bacteriophage are these viruses that infect bacteria. They use bacteriophage to show that DNA rather than protein is the hereditary material in a cell. Okay, so there was this debate at the time of whether DNA or protein, which one is responsible for passing on genetic material. On one hand, protein has like 20 amino acids, so it seems like it's able to encode for more information compared to DNA, which only has 4 different bases. And then, there were arguments from both sides until Hershey and Chase did their experiment with bacteriophage, and they were able to show definitively that it is DNA that's responsible for transmitting, encoding rather, the hereditary material, which will be passed on to the next generation. Messelson and Stahl, what they did is they proved that the model of DNA replication is, anybody remember what model DNA replication follows? Just to see if anybody knows. Okay, it is the semi-conservative model. Right, so there were 3 models at the time. There was the conservative model, there's the semi-conservative, and the dispersive model. So the semi-conservative model means that after DNA replication, you would have one strand that consists of entirely original material, and then you would have another double helix that consists of completely new material. That is the conservative model. And in the semi-conservative model, one strand would be completely original material, while the other strand is going to be new material. Okay, so I can draw this out for you. Let's say this is the original DNA. In the conservative model, what you will end up having is one double helix with original material, and then you will have a new double helix that's made up of entirely new material. This is the conservative model. In the semi-conservative model, you start with the parent DNA, And then afterwards, you would have one strand that is old original material and another that is new. Okay. And then there's dispersive model, which is like all mixed together. It doesn't have like each strand would have both new and old material kind of interspersed everywhere. And Meselson and Stahl, they show that it is the semi-conservative model that is the correct one. Any question before we move on? All right. Write the complementary strand for DNA strand below. Be sure to include directionality. Okay, so it's good to remember that DNA strands are complementary. That means if you know one side, you can predict the other side using Chagav's rule. And the other property is that they are anti-parallel. Anti-parallel. Okay. So to show for directionality, we use 3' to represent one direction, 5' to represent the other. Okay. So as a loose analogy to help you understand, you can imagine a bunch of people, standing on each other's shoulder or something. Very acrobatic people. And we will call this the head. The head, right? And then over here, that's the feet. Now, on the other side, we would have a bunch of people, but upside down. Okay, upside down. Okay, and then they will be holding hands to show the bonds. And this side will be feet to head. Okay, so they're essentially the same thing, same organization, but one side is head to feet, the other side is feet to head. Okay. Now, molecules don't have head or feet, so instead we use numbers. Okay, 3' to 5' on one strand, and then because it's anti-parallel, the other strand is going to be 5' to 3'. Okay. So what would be the complementary strand? Well, we know the base pairing rule. So A pairs with T, T pairs with A, C pairs with G, and then C, C, T, A, A, T. That would be the complementary DNA strand. Okay. Next, if 23% of all cells' nucleotides are adenine, what would be the percentage of thymine, cytosine, and guanine? Okay. So 23% is adenine. According to Chekhov's rule, we know that the percentage of A is going to equal to the percentage of T. So if you have 23% A, you're going to have 23% thymine. If you add these together, you will get 46%. Okay, 46%. Which means the percent of C plus G is going to be 100 minus 46, which will give you 54. Of the 54, half of it is going to be C, and half of it is going to be G. Okay, 27 plus 27 gives you 54. 23, 23 gives you the 46. Does that make sense? Any question about the calculations here? All right. So you're going to try a question for me. And I will see if you understand. Go ahead, please. Okay, anyone else want to try? Okay. Most of you got it right. So this is 33% are adenine. So 33% A, which means we're gonna have 33% T. We add these together, you get 66%, right? 33 plus 33 is 66. So if you take 100 minus that, you will have 34%, which are gonna be C and G. 37 divided by 2, 17. Okay. Don't forget to divide by 2. Questions? All right. The 4 stages of cell cycle, okay? What are they? Anybody? 4 stages of cell cycle. Can anyone tell me? Okay, so there is G1, there's the S phase, G2, and then there's the M phase. So M is for the mitosis, right? And then these G1, S phase, and G2, right? These here, these are part of the interphase. Okay, so a cell spends like majority of the time, like 90% of the time in interphase. And then when it's ready, it's going to undergo mitosis to divide. Okay. So the S phase is where you actually do the DNA replication, right? This is where you do the DNA replication. So before you do DNA replication, you got to check to see if your DNA is good enough for copying. And after you copy, you also have to check again to make sure you did not make any mistakes. Okay, so that's what the G1 and G2 are. They are called cell cycle checkpoints. Okay, so in G1, what do we check for? Okay, checked for the quality of DNA. So for example, any DNA damage that was not repaired, for example. You might also want to check for the length of telomeres. Okay. So if there are too much damage or the telomeres are too short, then the cell will not be allowed to proceed to the next phase. Okay. So if there are unrepaired DNA damage or telomeres are too short, then the cell will not copy its DNA. Okay, so you will not be allowed to duplicate that cell. Okay, if everything's good, then you will proceed to the S phase where you will duplicate the DNA. You will make a copy of all the DNA you have. And then afterwards, you will have another checkpoint, G2. Okay, so in G2, what you do is you check the copied DNA for errors and damage. Okay. So sometimes you made a mistake when you copy, you need to fix that. If it's damaged, you could try to repair it, but not everything is repairable. Okay. So what happens? If the cell fails G1 or G2, it might trigger. It might trigger apoptosis, okay, which is programmed cell death, or cell cycle, or like say growth arrest. Right? It's not going to be allowed to replicate anymore. Okay, so as you know, as we get older, our cells divide, but there is a finite number of times that they could divide. Once the telomeres are critically short, that cell would not be allowed to divide anymore, but not necessarily going to trigger apoptosis, right? Apoptosis is more for like DNA damage, but when the telomeres are critically short, they enter growth arrest. Okay, so that cell is not going to be allowed to divide anymore. Okay, we also call that senescence. Okay, so these are kind of like, sometimes they're called like zombie cells. They're just there in the body. They're no longer going to be able to duplicate, but they're not really dead either. Okay, and the buildup of senescent cells is part of the reason of why we age, why we get old. Any questions so far about any of the stuff that we've talked about? We all good? Okay. 4 stages of mitosis. What are the 4 stages of mitosis? Anybody? Anyone remember? What are the 4 stages of mitosis? Yep, there is anaphase. That's going to be like the 3rd step. Very good. Telophase. And then what's the other 2? Yeah, metaphase. And then, yeah, we didn't really talk about prometaphase, but you're right, Billy, that it does exist, but we just try to keep things simple here. And in front of that is prophase. Thank you, Savi. Okay, so remember, PMET. Okay, that's the order. Okay, someone wants me to go through the calculations again from earlier. I will do that afterwards. Okay, let's keep going for now. Okay, PMET. That's how you remember. All right, before we do mitosis though, let's fill in this chart related to all the enzymes that are involved in DNA replication. Okay, so first there is a gyrase. Gyrase, what does gyrase do? Gyrase is important to relieve tensions that builds up as the DNA is unwinding. Okay, so you probably know if you watch the recording that when you duplicate the DNA, you got to unwind the DNA, you got to open it up. Okay, but that opening is going to put strain on the DNA that is further down. And all the tension will build up. And if we don't relieve those tension, then the DNA will just snap off. Okay. Think of like a guitar string. Okay. If you tighten it too much, eventually it's gonna snap, right? So this is similar to that. So the function of the gyrase is to relieve the tension as the DNA is being unwound, and it prevents it from breaking. Okay, prevents it from breaking. Helicase. Okay, helicase. This is the enzyme that is responsible for unwinding the DNA. So it unwinds the DNA, the parent DNA, to create a replication bubble. It travels towards or into the replication fork. as DNA is being copied. Okay. So this is your double helix. The helicase will come and open it up to create a bubble. Okay, that's the replication bubble. Replication bubble. Okay. And then here is going to be your helicase. And it will travel into the fork. This is the replication fork as the DNA is being copied. Okay. Single strand binding proteins. Now, the DNA, of course, it has the nucleotides in it, okay, like A, T, C, G, or something. And on the other side, it would be the complementary base pair. So they are attracted to each other, right? The A is attracted to the T, the C is attracted to G, so on and so forth. And so what happens is they want to close up again after helicase unwinds it. And we don't want them to close up because we want to build new strands, right? And that's what the single-stranded binding protein is. And single-stranded binding protein will bind to the sugar phosphate backbone. Of the unwound unwind the DNA and prevent reannealing. Okay, that's just a fancy word for like coming back together. Okay, we don't want them to come back together. We want to keep them apart so we can copy them. Okay, next we have primates, okay, and DNA polymerase. Okay, let's talk about DNA polymerase first. DNA polymerase is the enzyme that will synthesize, or it will use the parent DNA, as a template to build the complementary strand. Okay. So again, let me try to draw this out for you. And let's say this is A, T, T, C, C, G, T, A, A, G, G, C. The DNA polymerase 3 will come and look at the parent strand. It'll be like, oh, this is a C. I know what to do. Let's put a G there. Okay, and then we'll look at the next one. Find the right nucleotide. Look at the next one, and so on and so forth. So in this way, it will build a complementary strand. Okay. Any questions so far? Am I making sense here? I hope so. Okay, so you don't need to write that down. Now, it's important to understand there are two limitations for DNA polymerase 3. Okay, number one, cannot start on its own. Cannot start on its own. It doesn't know where to begin. Okay, it's like a grade 3 kid come home. They don't know how to automatically start doing homework. Someone has to tell them to do their homework. Okay, turn to the right page, right, in the workbook and tell them do the homework. Okay, that someone is like a parent, perhaps. Okay, so DNA polymerase 3 is like that kid. Doesn't know where to start. Okay, needs someone to tell them where to go. And then second limitation is can only build in 5' to 3' direction. Okay. So how do we solve the problem? Okay, of it doesn't know how to start on its own. Well, that's what the RNA primase is for. The RNA primase is an enzyme that will create an RNA primer that will help DNA polymerase 3 to start the replication process. Okay. That's what's going to be happening. All right. How is that? Let's recap of what's going on for a second. Okay, if we go all the way down at the bottom, there is a diagram here. Okay. So we have the parent DNA shown here. Okay. Parent DNA. Sorry, it's a little bit hard to see in black and white, but that is the parent DNA. Okay, and this here is one side of the replication bubble. Okay, so like you create a bubble and then you're focusing on this area. Okay. Number 2 here, this enzyme, that is the helicase that I was talking about, right? Helicase. It is going to continue to travel into the fork to unzip more. Think of this as the, I don't know what you call it, like the thing that you pull on your zipper, right? So it just unzip your zipper. Okay, the helicase, the thing that you hold on to, it just keeps on unzipping your DNA. Can someone tell me what number 1 is, please? What is this thing doing here? What should we call it? Let's color it green. What is that? Thank you, KZN. This is the gyrase. Okay, and it's going to relieve the tension that builds up in the parent DNA as the helicase is unwinding. Okay. And like I said, the 2 strands that you pull apart wants to come back together. And we cannot allow that to happen. That's why we need these orange balls. What are the orange balls? They are the single-stranded binding protein, okay, which keeps the strands from reannealing. You don't want them to come back together. Okay. All right, this is again hard to see because it's black and white. I do apologize. But there is going to be like a little thing here. It's going to be another pink thing here. Okay, maybe another one here. Okay. And certainly, let's just draw another one here. Okay, so these pink nucleotides, those are the RNA primer. Okay. The purpose of the RNA primer is to tell the DNA polymerase 3 where to start. Okay, you should start here and keep on copying. What puts the RNA primer there? Well, you need another enzyme for it, and that's what this guy is, this pink guy. That is going to be the RNA primase. All these fancy names. Okay. Do you guys know, like, before I was teacher, I did research at SickKids, and there are these vending machines on the research floor, kind of like one of those snack vending machines, but instead of like potato chips coming down, you would be able to buy like RNA primase, or you would be able to buy like, you know, ligase, and all these little package on these vending machines. So you would insert the lab's credit card and you can buy these things to do experiments. So it turns out these things are actually quite useful in biotechnology, which is something we'll be learning in 2 or 3 weeks time. Okay, just one more thing here. One more thing. What is this little blue oval here? What would that be? Anybody want to help me out here? What does that represent? What does primase do again? Okay, so primase puts the RNA primer, right? And the RNA primer is needed to tell the DNA polymerase 3 where to start. Okay, remember DNA polymerase has 2 limitations, right? One of the limitations is that it does not know where to start. Okay, so by having the primer there, right, it's like start right here, start right there. Hope that answers your question. And that kind of gives this away. This little blue oval, this is the DNA polymerase 3. Okay, that's all we've talked about so far. All right. It's much better if you watch an animation of this, and it's available under the video resource that I post on eCentennial. Okay, fantastic. Let's go back to our chart and see what's going on. Next, we have the DNA polymerase 1. What does that do? Okay, so sorry, let me switch back to here for a second. Okay, so eventually, if you follow this DNA polymerase 3, right? Okay, so what it will do is that it will build new DNA strand, but eventually it's going to get closer and closer to the RNA primer that's ahead of it, right? Okay, so as it gets closer and closer to the primer, the DNA polymerase 3 will fall off, and then another polymerase called the DNA polymerase 1 will come in. And the job of the DNA polymerase 1 is to remove the RNA primer and replace it with DNA. Okay, because the RNA primer is RNA, right? So instead of having thymine, it's going to have uracil in it. Right? So an RNA primer might look like this. It might look like AUCCG, something like this. So the DNA polymerase 1 will come, and then it will remove the U and replace it with T. Okay. Let me see right here. So DNA is polymerase 1. Okay. It will replace RNA primer with DNA nucleotide. Okay. I'm just going to try to show you another thing. You don't have to draw this. Okay, this next part you can just watch. So we have the fork here, right? The fork. 3' to 5', and then 5' to 3'. So let's say you put the primer here. You put the primer here. The RNA primase will put the primer here, and then the DNA polymerase 3 will come. Right, I think it was blue on the other one. DNA polymerase 3 will come. DNA polymerase 3 will come, and then it will start building the new strand. Okay, remember the second limitation of DNA polymerase 3 is that it can only build from 5' to 3'. So it will only be able to go this way. Okay, remember DNA is anti-parallel. So if the black strand is 3 to 5, the new one you're building is 5 to 3. Okay, so on the other strand, same thing. There's going to be a primer. Right? This is 5 to 3. So this would be 5 to 3 this way. Okay. So what's the problem? Well, the problem is as we open up more and more of the DNA, how are we going to replicate this part? Okay. Well, you will need to have another primer and another primer, right? So then another DNA polymerase 3 can come and then make this part and then make this part. So for this reason, once on one side of the fork, one strand will be replicated in fragments. Okay, these fragments are actually called. Anybody remember what they were called? The fragments. The Okazaki fragment. That's right, named after the scientists who discovered them. Okazaki fragments. And because this strand is replicated in fragments, we call this the lagging strand. Lagging strand. On the other side, which is being replicated continuously, that would be the leading strand. So again, just to repeat myself because that's what teachers do. There is the another DNA polymerase tree here, but as the DNA polymerase tree gets close to the primer here, it will fall off, and then the DNA polymerase 1 will come in and replace the primer. Okay, replace it with DNA. Okay? But something has to glue it together at the end, and that's the function of the next thing on your table. They are called a ligase. Okay, but before I go back to the table, I also want to mention something else. Okay, let's say this is the end of a DNA, right? Okay, so as it opens, you're gonna have an other. Let's say you're gonna put an other primer here, and then you're gonna eventually copy this. Okay, remember, all these primers are RNA, and they will be replaced with DNA because of polymerase 1. Is that point clear? Just want to make sure that you understand what I'm saying. Okay, all these are RNA, and they will be replaced with DNA by DNA polymerase 1. Is that okay? I don't see any question. I'll assume that's okay. Here's the problem. Okay, if you look at the very end here, there is an RNA primer, but there is nothing coming from upstream. There is no DNA polymerase coming from this way. So after you finish copying the DNA, we will lose this chunk of the DNA. Okay, so if your DNA is this long, every time you divide, you lose a little bit, and you lose a little bit, and you lose a little bit. Okay, eventually you will lose important genetic information, right? So that is not good. We don't want that to happen. That's why located at the ends of your DNA, we're gonna have a bunch of, probably shouldn't use red, it's confusing. We are going to have a bunch of repeating DNAs at the end. Okay, it's like TTA, GGG. Okay, and they repeat it like, I don't know, hundreds of times. Okay, so every time you do DNA replication, you will cut off a little bit of these repeats. You will cut it off a little bit, and those are the telomeres. So instead of cutting into the important genetic information shown here in black, you will only be losing the telomeres. Okay, so if you divide again, oh my goodness, if you divide again, you will get shorter and shorter telomeres. Okay, so after many divisions, depending on the cell type, like maybe 30 times or 40 times, the telomeres are going to be critically short. Okay, and that's a signal that tells your cell, if you divide one more time, if you try to divide one more time, you will lose all the telomeres and you will start eating into the important genetic information. So I suggest you don't divide. Okay, so that's when the G1 checkpoint is going to kick in, and it presses on the brake of the cell cycle. It says, don't divide anymore. Does that make any sense? Any question? Okay. Ligase. What are we talking about here? Ligase. So ligase is the enzyme that will connect, right? Like ligate, right? That's what ligation means, to connect together. So connect the Okazaki fragments together. And the Okazaki fragments are on the lagging strand. Okay. Telomerase. Not every cell expresses telomerase, but in stem cells, which are cells that can become any cell type in the body, in stem cells, in germ cells, which are cells that will become sperm and eggs, we have an enzyme called telomerase. And the function of telomerase is to add TTA GGG to telomeres. Okay, in stem cells, and germ cells. Again, germ cells, many people think like germ cells are like bacteria because we hear like germs, right? But germ cells are actually cells that would become sperm or egg in the human bodies. It's important to re-lengthen the telomeres, right? Specifically in germline. Okay. Otherwise, when you have a kid, then you will be passing on chromosomes that have short telomeres, right? And then each generation will get shorter and shorter, and then eventually we won't be able to have kids anymore, right? So in the germ cells, we would reset the telomeres, and then the kid is born with fresh new telomeres. Okay, and then when they divide, they would shorten their own telomeres instead. That's it for all the enzymes and stuff. Are you sure nobody has any question? Fantastic. Okay, let's do another poll. This one, I mean, I didn't really talk about it like now, but you know, you probably, if you watch the recordings, you probably remember some of these things that I said. We'll do it anyways, okay, and then I'll take it up with you. Okay, just a couple more minutes just to see if anyone else wants to participate. Okay. So prokaryotes are like bacterias, right? And eukaryotes are like us. The main difference between prokaryotes and eukaryotes in terms of DNA is that for prokaryotes, their DNA is circular DNA. Okay, so it comes in a ring. And for prokaryotes, our DNA are linear DNA. Okay, because it's circular, they don't have the problem of eroding the ends of a DNA every time they replicate. Because let's say they put a primer here, right? Okay. And then they go replicate, it will just start here and then it will go full loop and then it will go back on itself and take these things off, right? Whereas if it's linear, we have that problem that we were talking about, right? You have a bunch of primer here, and then you go copy your DNA. Okay, nothing will replace this end, right? So that's why it's eroding the ends every time you divide. So we need to have the telomeres to protect the end. Prokaryotes, they don't. So prokaryotes actually do not have telomeres. It is the other way around. Okay, the other way around. Prokaryote chromosomes have a single origin of replication, whereas eukaryotic chromosomes have many. That is true. Okay, our DNAs are very, very long. So rather than having a like if you think about the DNA like this, okay, very, very long, and you only create like one replication bubble like this, it will take forever for the bubble to expand all the way to the end. So instead of doing this, you will open multiple bubbles everywhere, and then it will be able to copy much faster. Histones are proteins that package DNA. Histones, they package DNA. Prokaryotes, they don't really have a lot of DNA, so they don't really have to package it too much. Eukaryotic cells, we have tons of DNA. We must package it, so we do have histones. And finally, eukaryotes have the Okazaki fragments. but not the prokaryotes. Okay, so the answer is B. And so now you know. Now, if you see this question on the test, you will know what to choose. All right, now the main event, the mitosis. And let's try to look at this diagram. Okay. Actually, you know what? I want to clarify a few terms before we go on. Okay, so there is okay. First of all, there is DNA. There is chromatin. There is chromosome. And then there is also sister chromatids. Okay. Anybody want to attempt to explain the difference between some of these terms? Anyone at all? You can unmute. I invite you to speak. Or if you feel more comfortable, you can leave the answer in the chat box. I'm going to wait a little bit, see if anybody want to explain. Don't be shy. Okay, well, maybe you explained it in your own mind. That's also good exercise. But here is the difference, okay? DNA is just the macromolecules, right? So chromatin is actually DNA plus packaging protein. Packaging proteins. Okay. E.g., the histones that we talk about. Okay. So together, it will cause them to coil up. Okay. Coiled up. Okay. Otherwise, there's no way we can pack all these things into the tiny, tiny nucleus. So the DNA plus the protein gives us the chromatin. Okay. Protein, chromatin. Okay. That's how I remember it. Now, when you want to move the chromosomes around during cell, when you want to move the DNA, I should say, when you want to move the DNA around during cell division, and they're in this form, they tangle easily. Okay, so we don't want that to happen. So it will condense more, condense more to form the chromosome. So this thing we're going to have more packaging protein, and then it's going to look like this. It's going to package even more. Okay, again, you don't have to draw this necessarily. I'm just like making stuff up as I go. Okay, it's really hard to follow, I guess. Like that. Okay, and that's how we get the chromosome. And Safi says in the chat box, chromosome is the more condensed version of chromatin. That is exactly it. Okay, these are the exact same thing, but one is more condensed than the other. It's easier to move. Okay, this is like all your clothes, like just in a big pile on the floor. Okay, this would be like the same big pile, but then like stuff into like a suitcase or something. Is easier to carry around, easier to move around. Okay. So what happens is, let's say you have one chromosome. Okay, one chromosome. I'm just gonna draw. You know, this is basically one chromosome. Okay, and then after DNA replication, you're gonna have the exact same copy, and they will be stuck together. Okay, these are identical copies of each other. Only identical copies will stick together, and these are called sister chromatids. Okay, sister chromatids. They are genetically identical copies. Okay, so basically, this is what happened. You have one chromosome from mom, one chromosome from dad. But technically, you have 23 from mom, 23 from dad. Okay, but just focus on one of them. Okay, chromosome 1 from mom, chromosome 1 from dad. Okay, and then what happens is DNA replication happens, and now you have 2 of each. 2 chromosome 1 from mom, 2 chromosome 1 from dad, and then they're stuck together at this point, right? The centromere. So this copy and this copy, they're exactly the same. We call them sister chromatids. Okay, sister chromatids. So there you have it. Those are the difference. Hey, let's look at this picture. Identify and briefly explain the function of each of the cell structure. A is pointing at the chromosome, I believe. Chromosome. What that does is it contains genetic information. Okay. B, each chromosome has like a specific location that they like to stick to after they replicate. Okay. So, usually it forms like an X. So this location here, that's the centromere. Centromere. Okay. It is where duplicated DNA sticks together. Stick together. Okay. And for those of you who care about this stuff, the shorter part, that's called a P arm. And then the longer arm, that's called a Q arm. Okay. So if later on we'll learn like the location of a gene. You hear people say this, right? Like they found a gene that is tied to, early onset Alzheimer's, and it is on like, 4P, whatever, right? That means it's on the P arm of chromosome 4, there's a particular location, and that's where the gene is. Okay, C, anybody can tell me what C is? C is centrioles, that is correct. Thank you. They produce spindle fibers. Okay, produce spindle fibers. D is just cell membrane. Not sure why that is even there. Okay. Nothing to say. And E, that would be the spindle fibers. Spindle fibers. They will guide chromosome movement. All right. That is that. Any questions? Okay, we're going to take a break soon, but someone asked me to go through the calculation thing again, so I'm going to quickly do that. Let's just try another one. Okay, let's say we have 20% C, okay, and then we're going to figure out the rest. Okay. So C equals the G. So if you have 20C, you're going to have 20G. That's the first step. Okay, you figure out what's the complementary nucleotide, that's going to have the same percentage. And you're going to add them up. So it's going to be 40%. Right? 20 plus 20 is 40%. And then this number, that number is going to be 100% minus whatever you had for the other two. So if C and G collectively is 40, the remaining nucleotide makes up 60% of all the total nucleotides. Okay, so the remaining nucleotides are T and A. Right? And because A and T are the same, we would take 60 and divide by 2, which gives me 30 and 30. Okay, so these numbers is 60, 60 divided by 2. That's how you do it. Okay, does that answer your question? The student who asked this earlier. Good. Okay, 5 minutes break, and we will come back and do more. See you soon. Hello again. Let's keep going. Complete the following chart related to cell cycle. Okay. So again, interphase is where the cell spends most of its time. Okay, it's going to be performing its normal function. It's going to be growing in the interphase, going to be acquiring nutrients. Okay, and then in the interphase, that's where you will also have the G1, the S, and the G2 phase. Okay, so if all goes well, by the end of the interphase, okay, the following will happen. Okay, we'll call this the end of interphase and maybe like beginning of prophase. Okay, because it's hard to draw a boundary. It's like one transition seamlessly into the other. Okay, but basically what's going to be happening here is that in the end of interphase and the beginning of prophase, DNA replication is completed. Okay, and the chromatin, which is all these like mumbo jumbo stuff here, all this tangled stuff, starts to condense into chromosome. And then nuclear envelope starts to dissolve. And then by the time we finish with prophase, or like when we actually add the prophase, the nuclear envelope would be completely dissolved towards the end. Okay? And the centrioles move to opposite poles of the cell and start to produce spindle fibers. Spindle fibers are microtubules, right? That we talked about from like way back when, lecture 1. Okay. Sister chromatids are held together at the centromere. Okay. That's basically what happens in prophase. Metaphase. M for middle. You can remember that. M for middle. Middle. Okay. P for prepare. It's preparing to split the cell. So here, the spindle fibers attach to the centromere of each sister chromatid pairs and align them at the center of the cell. Okay. So you line them all up in the middle. That's what metaphase is. Next, we have the anaphase. Okay. A for apart. Okay, that's how I remember it. The sister chromatids are separated, pulled apart, and each will move to the opposite pole of the cell. Okay, give you a moment to catch up if you need to. And T for tearing apart. Okay. Now that the chromosomes, so like if this was a human cell, then you would have 46 going this way and 46 going this way. Now that they're at opposite ends of the poles, you will be able to separate a cell. Okay. So telophase is basically everything that's going to be happening. It's going to be the backwards of everything that happened in prophase. So over here, you will reform the nuclear envelope. You will take the chromosomes uncoil back into chromatin. Okay. Spindle fibers disappear. And then you have cytokinesis, right, which is separating the cytoplasm to create two cells. Okay. So this here, it's called a cleavage furrow. It's going to go all the way through the cell and cause them to separate. Okay. And that is essentially the telophase. Any questions? It's pretty straightforward, right? I hope. Hey, let's do some polls. There's one. Go ahead. All right. So a cell says 8 chromosomes at metaphase of mitosis. How many chromosomes would it have during anaphase? The answer should be 4, right? Okay. Because remember, metaphase, they all lined up. Okay. So that's 1 chromosome. My sister chromatids. 1, 2, 3, 4, 5, 6, 7, 8. When they separate, you will have like, 1, 2, 3, 4. Right? They will get pulled to the opposite side, right? Okay. So that is that. Okay, let's try the next one. Yeah, most of you got it. It is the telophase. Very good. Okay, here is another question. It's not a poll, but it's something to think about. Okay. I have trouble saying this word, but I think it's paclitaxel, something like that. It's a drug. It inhibits the formation of spindle fibers. Spindle fibers. Explain why it is used as a chemotherapy drug for cancer. Okay, so go ahead and let me know if you have any insights. Okay. Again, I welcome you to explain if you want. Anybody? Okay. All right. Safi said it stops spindle fibers so the cell cannot divide. Yeah, that's basically it, right? If you don't have spindle fibers, the cell would not be able to move the chromosomes. And as a result, you will not be able to divide. And that's what you want for cancer treatment. Okay, you want to stop uncontrolled cell division. Okay, so the chemotherapy drug is going to do just that. And the side effect is the drug might have off-target effects. It might stop the division of other cells. Okay, so you might have heard of people who are undergoing chemotherapy. They might lose hair, right? Because of limited cell division. They might have a weakened immune system because the bone marrow is being suppressed. So those cells are not creating new blood cells. Their immune system is going to be weakened as well because you're not getting new white cells. So those are some of the potential side effects. Exactly. Okay, blocks cancer growth by stopping cell division, resulting in cell death. Good, good. Okay. Let's see. I think that's enough polls for now. All right. So long answer questions. Any requests here for question 1, 2, or 3? Number 2. Okay, sure. We will do number 2. Okay, let's talk about this one. Oh, you know what? I just realized something. Sorry. This question that we just did here, I understand why you guys chose 16. And I think, you know what, you are actually right. Sometimes I get confused about this thing myself. So I apologize. Let me explain this again. Okay. Okay. So this is one chromosome, yes? One chromosome. One chromosome. Okay. If you make a copy of this, then now it's like this, right? And they're stuck together at the centromere, right? Okay, this is important. Please listen carefully. So after DNA replication here, if they're stuck together, how many chromosomes do we have here? I thought this was 2 chromosomes, but I actually remember now. Okay, this is actually still 1 chromosome. Okay, because they are like stuck together, we still count this as 1. Does that make any sense? It doesn't really make sense to me because I thought it's 1 and 2, but I forgot the very important part that if they are stuck together, that's considered as 1 chromosome. Is that okay? Do you guys understand what I'm saying? So that's 1 chromosome. And after you duplicate, it is still 1 chromosome. Is that okay? Give me some response here. Okay. Thank you, Christian. Christian is always letting me know that things are okay. Thank you. Okay, so if that's the case, and then it says the cells has 8 chromosomes in metaphase, so that means we have 8 of these things. 8 of these things. So 1, oh my God, this is hard to draw. 1, right? And then 8 of it. So let me just copy and paste. 1, 2. Does this even work? Okay, 3. Oh, this is horrible. Okay, let me copy both of them. Okay, so that's like 3 and 4, right? And then let's just copy them one more time. Oh, there we go. There are 8. Okay, 1, 2, 3, 4, 5, 6, 7, 8. Okay, and then when they separate, let's say when this one separates, then one of the sister chromatids get pulled this way, the other gets pulled this way, right? So that's 1, 2, and then same thing. 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So after they get separate, then they no longer count as 1. So that's why it becomes 16. Okay, 16 is the correct answer. If you see this question on the test, choose 16. Okay, 16 is the correct answer. Does that make sense? Sorry, I just got confused. Yes, I know. A lot of you chose 16 and I said you were wrong and I'm sorry. Okay, my bad. You guys are correct. 16 is the correct answer. 4 is not the correct answer. Choose 16. Okay, that's all I can say. Again, I apologize for that. Let's go back to this very exciting question. Answer the following questions related to Michelson and Stahl's experiment. I'm going to copy this and put it on a new page. Okay. Answer the following questions related to Michelson and Stahl's experiment. What is the purpose of Hershey and Chase experiment? Okay. So I can write down for you the purpose is to determine the model for DNA replication. Okay, the model for DNA replication. Okay. Is it semi-conservative? Is it conservative? Or is it dispersive? Okay. B. Maybe I should write down. Is it conservative? Is it semi-conservative? Or is it dispersive? Okay. Let me pull up the PowerPoint rather than me drawing it. It's probably going to look a little bit nicer. Give me a second. This is lecture number 6. Okay, so let me explain using this picture. It's much easier that way. Okay, so in the semi-conservative model, we start with a parent DNA. Okay, so shown here in pink. And then the semi-conservative model says that after one division, here are your daughter's DNA. Daughters doesn't mean female, right? It just means like, you know, the afterwards, the 2 new DNA, what do we call them? We call them daughter's DNA. Somehow the biology pronoun is feminine. So one strand would be made up of original material, shown here in pink, and then the other strand is made up of entirely new material, shown here in blue. Okay, so blue, like the blue and pink, like this. In the conservative model, you will have one DNA molecule made up of entirely new material, shown in blue, and one DNA molecule made up of entirely original material. In the dispersive model, it's both strands of the daughter DNA consists of new and old material. Okay, so the difference between this one and this one is that the old material is restricted to one strand. Okay, whereas the old material here is dispersed between both of the strands. Okay, so that's what the both of the model is. Okay, we'll take a look at the second generation in a bit. What is the difference between N15 and N14? So they are basically isotopes of different weight. Okay, can someone tell me what is an isotope? Did you guys? Oh, thank you, Safi. You answered my question before I asked it. N15 is heavier, okay, than N14. An isotope is basically two atoms of the same element that has different number of neutrons. Okay, so by having an extra neutron in N15, it is heavier. Okay, so why is it important to grow the bacteria in N15 for several generations before transferring them to N14? Okay, so part C, growing them in N15 for like several generations ensures all the DNA starts off with N15 in them. Okay, starts off with N15 in them. So if I go back to this diagram, right, if these are like E. coli that I've been growing in N15 for several generations, then all these DNA will have the heavy version of the nitrogen. Okay, so if I extract the DNA and I put it in a test tube and I spin it down with a centrifuge, what's going to happen is because the N15 is heavy, right? Then it will sink to the bottom. So you will have like an N15 band that represents the DNA. And that will be the case for all of these samples. Okay, you're going to have an N15 band at the bottom. Does that make sense? So generation 1, all of them is going to have a heavy band at the bottom. So now I'm going to transfer them to the N14, which is lighter. So any new DNA that they're going to make after the transfer will be using the lighter N14. No more N15 will be supplied. Okay. So if the experiment, if the model is semi-conservative model, then when you extract the DNA from the first generation, you would expect to have like a medium band in the middle. Okay, this band is going to be a combination of N15 / N14. Okay, N15 / N14, because half of it will be the original material containing the N15, and the blue would be containing the N14. So you will have a band in the middle. If it is a conservative model, let me just move this out of the way. If it's conservative model and you collect the DNA and you spin it down, now you would expect to have two separate bands. And you would have a lighter N14 band corresponding to this completely new DNA. And you would have a N15 band in the bottom. Okay, if it's the dispersive model, then also you will have a mixed band that is in the middle, a mixed band. Okay, which is why the next question asks, why do we have to do it for like 2 generations, right? Why do we have to do it for 2 generations? Because if you only do the experiment for 1 generation, okay, and that's what they did it for 1 generation, and then they observed this result. Okay, they have a single band in the middle. Okay, not 2 bands, but 1 mixed band in the middle. There is no way at this point to tell whether it is the semi-conservative model or the dispersive model. So they do it for another generation. They allow the E. coli to grow in another generation with the with the N14, and then they extract the DNA. So you can see here, right, in the semi-conservative model, you would expect to have what kind of band? And so there's going to be a lighter band now, right? And then you're going to have a mixed band. Okay. Whereas for the dispersive model, you will continue to have a mixed band. So this is the result that was observed, and that's how they show it is the semi-conservative model that's correct. Any question about everything I just explained? Okay. Moving on. Application question. Answer the following question based on the karyotype. A karyotype is an orderly arrangement of chromosomes, right? So this is a karyotype. Which chromosome is the longest? Well, the longest chromosome is chromosome 1. Longest. And then the shortest is chromosome 22. How are chromosomes numbered? Okay, based on length and location of centromere. Okay, so there's the centromere for number 2, there's a centromere for number 3, centromere for number 4. It takes like a trained person to do these quickly. Okay, I wouldn't be able to organize it if it's all like mixed together. Okay, but someone like a cytologist, they would be able to do it. Does this karyotype belong to a male or female? Anybody know? Male or female? Female. How do you know that? Okay. Safi and Casey say it's female. Anyone else? Or does everybody think this is a female? Okay. XY. XY is male. Okay. XY is male. Okay. Female. Female is XX. Okay. It's the other way around. Yeah. But that's okay. Now we know. Okay. The Y chromosome is what makes the male male. Okay. Did you know by default all embryos are going to become female. All embryos are going to become female by default. But if there's the Y chromosome, there's a gene on the Y chromosome called the SRY gene. It gets turned on during embryonic development, and that causes the embryo to continue to develop as a male. So by default, they are going to turn into female embryos unless the Y chromosome is present. Explain whether the karyotype shows any abnormalities. Does anybody see anything strange in this karyotype that shouldn't be there? Oh, how can you tell which one is X, which one is Y? I mean, you cannot really tell, but then it says right here, right? It says X and Y, right? That's how you can tell. If I don't label it, then you can't really tell, okay? Other than the fact that Y is a lot shorter than the X, okay? If they're side by side, if I just show you these things and I tell you there is sex chromosome, then you know the Y is a lot shorter. Okay. Any abnormalities? Anybody? Anything strange on this karyotype? Very good, Makia. There are 3 copies of chromosome 21. Okay, so we call this trisomy 21. Okay, and that means Down syndrome. So not good, right? The extra chromosome 21 is going to cause various developmental problems. So that's one of the things that a karyotype can pick up. All right, fill in the blanks. Here we go. The 2 strands of DNA run in parallel but opposite direction. This property of DNA is called anti-parallel. DNA replication follows we just talked about it, the semi-conservative model. The longest phase in cell cycle is the interphase. That's when the cell is doing its regular job, its normal function, and growing, gathering resources, doubling its DNA. Following DNA replication, the sister chromatids are held together at the centromere. Something are short DNA segments synthesized discontinuously. That's the Okazaki fragment. The something strand is synthesized continuously during DNA replication. If it's synthesized continuously, it is called the leading strand. On the other side, where it is synthesized in the Okazaki fragments, that's the lagging strand. Something is the enzyme that uses the DNA, the template DNA strand to synthesize DNA from 5' to 3'. That would be the DNA polymerase 3. And then the DNA polymerase 1 is the enzyme that will replace the RNA primer with DNA towards the end of DNA replication. To create the primer, we need the primase. The TTA-GGG that are found at the end of chromosomes, those are called telomeres. Something is an enzyme that lengthens telomeres during gamete production. The telomerase. Okay, again, not in every single cell, right? You only have telomerase being expressed in germ cells, which are cells that will become sperminae or stem cells. A cancer actually turns on telomerase so that they can keep on dividing forever and forever. Okay, so one of the targets of cancer therapy is to suppress the telomerase. Sister chromatids line up in the middle of the cell during metaphase. A karyotype is a picture of an orderly arrangement of chromosomes. Something produces spindle fibers during mitosis. Centriole. Something happens at the end of telophase. Cytokinesis. Okay, let's fill in the diagrams here. Okay, some are plant cells, some are animal cells. Okay, so the first one here, I don't see any chromosomes at all. It just looks like a cell, ordinary cell. So that's probably in prophase. What's the next phase? Anyone? Anybody want to help me out with the next diagram? Telophase is correct. Yes, telophase is very distinctive. You can see its cell is splitting. This is, oh, I'm sorry. So I'm making so many mistakes today. This I meant is interphase. Because it's not doing anything yet. I don't see the chromosomes. I don't see any spindle fibers, nothing at all. So it's just interphase when it's just doing its own thing. This is the prophase. This is the prophase. Okay, what about the next one here? It looks like the chromosomes are being pulled apart. Right. So apart is anaphase. Yes. Okay. and other prophase here. And you can see the plant cells don't have any centrioles. Okay, the spindle fiber just kind of appear. Next one. This is anaphase. This is metaphase. And other metaphase. In the plant cells, you can see the 2 nuclei. So that tells you it is telophase. And then here is another anaphase. Okay. That is it. So I do want to quickly talk about something with you before I say goodbye. Let me see, screen 1 right here. So if we go to the course homepage for a second, I mean, you don't have to do it. I'll just show you. Course homepage. Okay, and let me see where the calendar is. Calendar. Right here. Okay, so we are in week 8 now. By the way, next week's lecture is complicated, so make sure you prepare accordingly and then come to the tutorial. We are going to be doing the transcription translation and with mutations and everything like that. And then quiz the week after, and then we're going to have a second lab. Okay, so I mentioned to you that the first lab was not done as well as I hoped you would. And so I want you to start working on lab number 2 like earlier rather than later. Okay, the lab is available now, right? You can download it and you can do it. So under content, you will see the virtual labs and then lab 2 microscopy. This is what you will have to download and fill in and submit. Okay, so once you download it, it looks something like this, and all the instruction is here. So first you have to watch the video, okay, and then you're gonna click on this link, which will take you to this virtual microscope. Okay, so in a classroom, I will show you how to use the microscope. Right, we will actually be preparing slides and stuff like that. Obviously, we cannot do that in a virtual setting. So the next best thing is just a simulation. I know it's not really the real thing, but that's the best we could do at the moment. And there are like a bunch of different slides here. Okay, animal slides and plant slide, and you would have different mitotic phase. So if I put one in, and then what happens is you will play around with it to see how to focus it and stuff like that. Okay, but basically there is a cell right there. Okay, there is a cell that is undergoing what phase is this? What phase did I say? Telophase, right? Anaphase, right? Okay, so you can see the chromosomes are being pulled apart. You can focus it a little bit more. So the lab will ask you to examine some of these slides, okay, and then you will have to take a picture of these things and then put it in the lab. So if you are using Windows, there is something called the snipping tools. Okay, you just type it in the what do you call this thing? The magnifying glass, and then you can click on New, and it will allow you to capture anything on your screen. Okay, so if I say that, then I will capture this picture. All right. So when you capture it, I don't want you to capture like the entire screen like this and then paste it into the document. It's too big, right? Okay. You can just copy only the parts that we need. Okay, and then afterwards, you should be able to do it with the window key, Shift, Window, and S. Okay, thanks for the tip, Christian. That's another way to do it. And then you will go to the Word document. Okay, I highlighted all the questions because last time people missed missed some of the questions, right? So I highlighted all of it this time. Hopefully it will be okay. And here it says, take a screenshot of a cell in anaphase. I know it says onion root. Okay, so like you would have to find the onion root, but you know, I will paste whatever picture I screenshot, and then you would make it smaller so it will fit. Okay, so don't use don't send me a link where I have to go to like your Google Drive or OneDrive or whatever to see the picture. You should copy and paste the picture directly into here. Okay. If you use Mac, you can search online how to do a screenshot. If you're not sure, you can email me. I can look it up for you. It's probably, if you just type it on Google how to screenshot on a Mac or on Apple, they will tell you. Okay. Also, the simulation here, I don't think it runs really well on the phone. So try to do it from a laptop, from a desktop, from a computer. Okay, rather than on your phone. Worst comes to worst, if you cannot figure out how to screenshot, you can just use your phone, right, to take a picture of the screen. Okay. Again, when you take the picture, like make sure you put the phone like right onto the actual diagram. Okay. Like in the past, I have people who take picture with the phone like all the way from here, like, and then they paste it in. So I see their table, I see their pizza. I'm not making this up. I literally saw pizza in one of the assignments, and that's not what I want. Okay, I don't want to have to zoom in to see the picture. Okay, you should just crop it and put it in. Okay, now the lab, it's pretty straightforward. Okay, there are some like parts that are a little bit confusing with the calculation and things like that, but I've created a YouTube video. This is actually me showing you how to do the calculation. So if you watch that, you should be able to figure it out. Okay. So here it says upload a picture or type your calculations here. Same thing. Okay. It's hard to type math symbols. Do you guys like write your math tests and then send a picture to your math teacher? Is that what you guys do? Yeah, so you would know what to do here. You write the calculations and then you put a picture in here. Okay, again, crop it, make it nice. But you can type it in as well. Okay, if you want. Okay, if you want to type in like, you know, whatever, 200 divided by 50 equals 2. You can do it like that as well. Okay, but writing is probably easier. So there's some calculations, and then there are some theory questions as well. Okay. Oh, thank you, Kia. The command on the Mac is Shift Command 3 on Mac. I don't know what that means. I guess there's a Shift key and then the Command key. I think there is a thing that looks like this, right? On Mac, something like that. That's the Command key and then 3. Right? That's how you do the screenshot. Thank you for letting us know. But yes, please do the work, do the lab early. If you don't understand what the question is asking, email me for clarification. And look at the marks here, right? It says 3 marks, right? What is the relationship between the image and the object? And then besides whatever gives 2 other letters. So you answer both questions to get 3 marks. Okay, and limits to 3 or 4 sentences. So keep it short, keep it concise, keep it correct. Okay, so please work on that. And 10% is gonna go a long way. It's actually gonna boost your grade significantly if you get 90% on something like this. Okay, so that's it for this week. Thank you very much for coming. Have a fantastic weekend, and I'll see you next Friday. Bye for now.